So I am going to tell you about what part of my brain occupies part of my brain, a large part of it, is about the nucleus. And I've been interested in epigenetics for a long time, and my lab has been focused on the cell biology of the nucleus. And we have kind of three questions that we're asking. We're trying to figure out how nuclear organization and architecture affects how chromosomes are organized and how the genome functions. But it's a reciprocal kind of relationship in that we're also trying to figure out what the genome does to the nucleus as an organelle, right? In particular, what epigenetic or chromatin level higher order organization of the genome may do to the nucleus in terms of its structure and how it behaves as a real organelle in the cell. And we're also interested in evolutionary cell biology questions because if you look at the different kingdoms of life, fungi, animals, plants, they share many of the same proteins and processes that, quote unquote, build a nucleus. But then each kingdom has their own kingdom-specific sets of proteins as well. So there's been different evolutionary paths that they've taken in these different kingdoms, and we're trying to figure out what the significance of that is. And so in terms of systems, mostly we think about plants. I've done most of this in Arabidopsis and more recently in tomato. And we're also doing some things in algae, and in particular streptophytic algae, which are sister groups to land plants. And then I have insects. Did I say insects? Yeah, okay. Insects, right? So animal cells, but most of this work that is done and understood about nuclear cell biology is done in mammals. So we're trying to use insects, partly because there's a tradition of insect cell biology here at BTI, but there's also some interesting characteristics in terms of genome variation. Okay, so we're doing genetics, some computational things, and some cell biology. So I think in the moments I have left, I just want to throw out a couple of things. All right, this beautiful picture is developing neuron, right? And what's important here is the yellow box. That's the nucleus. So you get the idea. It's like, it looks like a laundry bag with a bowling ball in it, right? And that is, there's not a whole lot of structural integrity to the cell, but the nucleus is, it's hanging in there. It gives you the idea that the nucleus is kind of like a jewelry box, right? It's protecting the genome inside. This is the idea. And so people have been able to go in and actually push on that nucleus and torture it in various sophisticated ways and show that it is provides structural protection, but the situation looks a little different in plants, right? So in our world, the box is around the cell, right? There's a cell wall, quite stiff cell wall. And then the nucleus has this rather, I don't know, it's got a marginal existence. Can you see it at the bottom? It's kind of squished in there, right? It's squished in between the other organelles, and there's a central vacuole, right? And then what's not shown here is, of course, the dynamics. There's cytoplasmic streaming. And so if you look at nuclei just under the microscope from plants, their shape changes over time, and they are frequently have irregular edges. So kind of intuition tells you, well, maybe this is not like a jewelry box at all. Maybe it's more flexible, right? That's intuition. But what we're trying to do is figure out if that's true or not, right? And so when I say we, I have a collaboration with Jan Lammerding's lab, and Noam and Jacob are members of Jan's lab who have been helping with all of this work. Alex, here in the audience is the glue between me and Jan's lab, who's been spearheading this. And I'll tell you about the Abbott lab in a second. But just in terms of techniques, because I don't think I've ever said this out loud here in this context, there's 3 things that we're trying. Most of these assays are set up for animal cells, and we're trying to adapt them for plant cells. And of course, there's that cell wall. When you take the cell wall off, you have this really fragile protoplast, and it doesn't want to stick to anything. There's all kinds of technical issues here that we're trying to deal with. So one approach is a relatively simple one. If you can get a cell to sit on a surface, you can push on it. That's the nano indentation. And there are devices that you can measure the force, and under the microscope you can see how far you're pushing down the surface of the nucleus, and you can make inferences about the mechanical properties of the nucleus. There's another assay that Jan's group has developed, which is called a micropipette aspiration, but it's really just a torture chamber, right? So it's a microfluidics device, and you're taking a big cell and you're forcing it down a pressure gradient into a tiny little channel, okay? And you're doing this because it's a microfluidics device, multiple different times, but here's just an example. I don't know if this is going to show up. You don't want to see that? I don't know. Maybe this won't show up. Oh, yeah. So you see there's a cell that enters. In this case, it's an insect cell. Sorry, I'd like to do this on plants. Can't do that yet, but that's an insect cell. It enters the channel, and it's being pushed by a pressure gradient. And what you see in blue is a DNA stain. And it starts, right? The nucleus is starting to deform and going down this channel, right? And so this is a way to determine its deformability. And then, of course, if you've got controls, you can set this up in such a way that you can compare mutants or non-mutants or things that have been treated with chemicals. And so what we're trying to do is change things like chromatin modifications to see if this changes the behavior, the physical behavior of the nucleus. All right, so that's cool, right? I don't want to say this is cooler, but this is interesting, right? To me, all right. So something that we started recently is a collaboration with Nick Abbott's lab. He's in chemical engineering at Cornell and working with Pervil, a graduate student in his lab, to use synthetic liquid crystals. And so these are solutions of the solutes, I guess are in those green stacks there. You can cause essentially sort of a phase transition from disordered, either at higher temperatures or lower solute concentrations, to a more ordered matrix, that's the one on the right, where the molecules, the solutes, start to line up with each other and they form essentially an ordered array. This starts to put physical pressure on cells or organelles that are floating in that matrix, right? And so what's shown there, it looks like a red blood cell because Nick's lab did pinching of red blood cells and can measure with proper models the force and again, the biomechanics. All right, I'm almost done. And so what we're trying to do now is to see if we can do this with isolated nuclei in plants. So these are the kind of techniques that we're doing, and it's been fun, but plants are always throwing us some curveballs, so we're trying to get around some of those technical things. All right, thank you all.